Cancer is the second leading cause of death in the United States, only behind heart disease. In a normal year, right, COVID-19 wouldn't have been here, and unintentional injuries would have followed as third. So what is it about cancer? Why is it so deadly? What is the disease itself? And why don't we have really great cures for this yet? Why does the incidence of cancer continue to seem to be going up? In this video, we'll look at the answers to some of those questions, talk about the mechanisms of cancer, some statistics about cancer, and some novel or interesting treatments that might be on the horizon. Let's get started. So I want to start with some statistics about cancer. In 2015, roughly 91 million people worldwide had cancer. And what we find is that each year there are roughly 20 million new cases of cancer. And globally, around the world, about 10 million deaths from cancer each year. So how does this break down? It's actually interesting. It seems that males have a higher chance, although it's just slight, of getting cancer at some point in their lives. This chart looks a little skewed. We only go from 15 to 20 here on our axes. But as you can see, the incidence of cancer in men seems to be more. And when we look at the types of cancer that men versus females get later on, we might be able to elucidate on that point a bit. So, what is cancer? What's going on? Is this something you catch? Is it something that happens to you? Well, let's talk about the mechanisms, the underlying biological mechanisms that cause cancer. So before we can really understand this, we need to go back to biology class and learn a little bit about the cell cycle. So you may remember a picture like this from your biology class. Basically, the cell cycle is divided into interphase, all of this, and then mitosis. You may remember mitosis is just the splitting of the cell, so it goes from one cell to two cells. These other things, G1, SG2, this is just when the cell's growing, making more DNA, and then growing some more, doubling those organelles to split. This is really the important part, mitosis. That's where one cell divides into two. So, the process of cellular division, or the life cycle of the cell, is actually highly regulated. It has all of these checkpoints. So a checkpoint, if you think of it, I like to think of it as a red light, green light situation. Do you get the green light? Do you pass? Do you get to go to the next stage? Or do you get the red light and fail? So there's lots of checkpoints I want to point out. The first one here is in G1, right? This is a growing stage. And what we're looking at is the cell, does it have the right number of organelles inside? Are the growth factors activated? If it gets the green light, it goes on to the next stage to make that DNA. After DNA has been synthesized here in the S phase, we go over to G2. And now what we're looking at is basically, is the cell ready to divide? Is it have large enough cell volume? Can we go ahead and divide and replicate the genome? Well, then there's another checkpoint. That's at the end of mitosis. That's after the chromosomes in anaphase have split apart. So we're wondering, did we have equal distribution of those chromosomes? Now, believe it or not, this checkpoint system is really good for our bodies because we don't want a bad cell, a mutated cell, to go through mitosis and make more bad cells, right? If we got one bad cell, we can deal with that. But if one bad cells become two bad cells, become four, et cetera, we start to develop a tumor. We don't want that. So these checkpoints are a good thing in our body. So what's really going on here? We know that our cell cycle is highly regulated by these proteins called cyclin. So they are appropriately named, I think, right? It's a protein, ends in IN, that is part of the cell cycle. So these cyclin proteins, as you can see in this chart here, they go through various stages and they let the cell progress to the next stage or not. So What cancer is, basically, is when these checkpoints aren't working correctly. For several different reasons, these checkpoints have been mutated, or some genes have been mutating, allowing the cell to just completely go through the cycle over and over and over and over with no checkpoints. So basically, cancer is caused by a few major things happening. One, growth factors. If you think about growth factor, right? Break down the word. A growth factor is just a factor in your body, all right? They're often proteins, maybe some lipids, that tell cells to grow, or that tell them to undergo mitosis, to go through the cell cycle. So, occasionally, though, these growth factors get out of control. Perhaps there's a mutation. And I want to talk about two things that happen a lot. One is proto-oncogenes. If you break down the word, proto, like a beginning, like a prototype, onco means cancer, like an oncologist would be a cancer doctor. These are normal genes, right, related to growth factors, that become cancerous genes when they're mutated. So basically, a proto-oncogene was just at one time a normal gene involved in these growth factors that had a mutation which now is being turned on all the time, right? So if you think about in your body, there are different times you need to grow different things, right? When I was young, when I was a baby, when I was in the womb, I needed to grow an arm. I don't need to grow an arm anymore, right? There are times in your body where you need to grow different things, or you need to make new proteins. Cells need to divide. Well, sometimes those cells don't need to divide. But when the growth factor genes are in a mutated state and are constantly on, then this can lead to cancer. Well, are we doomed? Well, not necessarily. The body has evolved things called tumor suppressor genes, right? These are genes that make functional proteins which essentially are looking for these tumors, these cancerous things, and then if it finds one, it turns off cell division, right? So it's a tumor suppressor. We don't want that tumor growing. A famous one that you'll hear about a lot is called tumor suppressor gene P53. This gene is what stops tumors. Well, if there is a mutation in that tumor suppressor gene, then the tumor is no longer suppressed. This leads to cancer. So let me show you sort of how this works. Normally, in normal cell division, you have a normal proto-oncogene, which is highly regulated, right? And then you have these tumor suppressor genes, which normally work. Cell cycle nice under control. We get nice normal cells. However, if we have malignant, cancerous cell division, we have a mutated tumor suppressor gene and often a mutated proto-oncogene, where that the proto-oncogene has now gained a lot of function, so it's saying grow, 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 grow. And then the tumor suppressor gene has lost its function, and it's saying, oh, I can't tell you to stop growing. So this one says grow, grow, grow. This one can't stop the growth. Therefore, we get cancer. So cancer, as I mentioned, is essentially a failure of cell division control. So we mentioned gene P53 is a key role in that G1 checkpoint. And what we have found to this point is that all cancers have to shut down that gene P53 activity to survive. So as I mentioned, gene P53 right here, this is normally what it helps to do, right? It can stop the cell cycle or arrest it, right? Say, stop it. It can also repair DNA that's been damaged, right? If there's a mutation in the DNA. It can also help to control senescence, right? How long something lives. And gene P53 also, if there's a bad mutation, it can tell the cell to undergo cellular suicide, or we call it apoptosis. Well, here's the problem. If we've got a mutation and we've got an oncogene activation, these things inhibit, right, gene P53. DNA damage should cause P53, right? You see this arrow activates P53 to correct the damage. But if we have these proto-oncogenes that have kicked in and caused the cancerous gene, we're going to limit gene P53 from working. Therefore, these things won't happen. Okay? So, what are the biological characteristics of cancer? There are several. I'm going to focus on 6 that generally have to happen for cancer to occur. The first one is these growth promoter genes that we just talked about. There's generally a mutation where these are turned on, okay? So we call this sustained proliferation signaling. These growth promoters are continually giving the signal to proliferate the cell growth. Another one that I just mentioned is these tumor suppressor genes, right? So the growth suppressors from P53 have to be turned off. So normally this tumor that you see there getting out of control would be stopped. But if these growth suppressors are turned off, then this cancerous tumor continues to grow. Another thing that cancer does, it enables replicative immortality, right? So if you think of the symbol here that we use a lot for infinity, basically what we're doing here is we're saying maintain the chromosomes so that they never die and they can replicate forever to infinity. So that gene needs to be mutated and turned on. We also need to activate invasion and metastasis. Metastasis is a fancy word for the spreading of the cancer, right? So someone says, oh, this cancer has metastasized. That's bad because it has broken off generally and spread. We don't want it to break off because if you think about in your body, most cells, organs need to be anchored somewhere, right? We don't want our liver floating around through our body. We don't want certain cells, with the exception of maybe a few, floating through the body. So we have to turn off what we call anchor and density dependent genes. So if the gene that makes sure something is anchored and doesn't get too dense is turned off, then we can get these dense tumors forming that metastasize. And then I think one of the meanest things that cancer does is it induces angiogenesis. So if you think about this word, genesis, the beginning, angio, we typically think of like our blood vessels. So inducing angiogenesis would mean making new blood vessels. Cancer is so nasty that what it does is as it's growing, it says, oh, hey, I need more nutrients. I need more blood. So it tells the body to make more blood vessels to go to the tumor itself, right? That's really a nasty thing that cancer does. And then the last one is it needs to resist cell death. So I told you that generally, if things are working correct, a bad cell will undergo cellular suicide, right? And die. That's a good thing. We don't want bad cells to grow. But in cancer, these apoptosis genes are actually turned off. Okay? So there's a lot going on here. But actually, what we're learning more and more is that all of these mutations have to occur for really cancer to get out of control. All right, so what causes these mutations? So I just listed all these mutations that occur for cancer to happen. Well, what causes these hits? What causes these mutations? Some of them you can predict, right? UV radiation, certain chemical exposure to radiation, right? If you think about your X-ray tech, they're always wearing those lead vests. We now know that some viruses cause mutations. Cigarette smoke, that's a big one. Pollution in some areas where it's really bad can cause mutations that could cause cancer. And then age and genetics. We know the older you are, the more likely you are to develop cancer. We also know that some people have a predisposed genetic disposition to get certain types of cancer. So let me show you kind of how this works. So I'm going to pick cigarette smoke. It's been studied a lot. So what happens, we know, when you inhale certain chemicals, specifically the ones in cigarette, it damages and stops the repair process of some of the DNA in these cells. So you get the cigarette smoking, that hit causes some DNA damage in that cell. Normally, right, the cell with cellular suicide, apoptosis. But if the proliferation gene is turned on, that bad cell divides, comes into 2 or 3 or 4, and then over time with more mutations, that genome has become very unstable. We've lost gene P53, the tumor suppressor, and we get a cancer, right? So all of these things you see here can lead to that DNA damage, which then becomes cancerous. All right, so now time for some stats. What of these things I've listed do we know can be attributed to most cancers that we see? Well, it should be no surprise to you that tobacco use is the highest attributor, or you could say contributor, to cancer. The second, Helicobacter pylori, that's a bacteria that can cause some cancers in the gut. Alcohol use, Hep B and C, that's a virus. Poor nutrition. HPV, human papillomavirus, we know actually causes cervical cancer in women. There's actually a vaccine you can get against HPV if you were worried about that. Overweight, obesity, physical inactivity. The biggest one, though, is tobacco use. So if you are someone consistently worried about cancer, you know it falls in your family, you should not be using tobacco. Period. End of story, right? Maybe even alcohol use you might want to include. These are harder to control, bacteria in the gut, but these two things you can control for sure. All right, so let's look at some stats on what kinds of cancers are common. In men, lung cancer, most common, right? Smoking, pollution, we see in specific countries that are really dense, followed by prostate. Prostate cancer is pretty common in men once they reach a certain age. So that's when men need to start getting their prostate checked. Then you go to colorectal, stomach, sort of down the line. Now, this is number of cases in the men in the United States. And by the way, this was from 2011. But this is the number of deaths. So this really shows you the deadlier cancers, right? Liver was lower in the cases, but look how it shot up in terms of death. Liver cancer, very bad. Notice that prostate was number 2, but when we find that early, we can treat it so it goes down. One that I want to point out, I guess, is that pancreatic cancer isn't even on the list of common, but it is on the list for deaths, meaning pancreatic cancer is highly deadly. Okay, so what's the most common cancer per geographical region? I find this to be a very interesting chart. What we see over here in lots of Asia and up here, we see that lung cancer, mostly from smoking and pollution, is the main cause of death. We go over to the Americas, and we see that prostate cancer is the most common cancer in men. Okay, so these are men. And then things really change. Africa has a lot of very different types of cancers that are the most common in those geographical regions. So let's go to women. As one would suspect, the number one case of women cancer in the United States is from breast cancer. Breast cancer, we see over and over many, many women getting breast cancer. And we know that breast cancer, as you see over here in the deaths, is also the number one deadliest cancer for women. Not because it can't be treated, because it can, right? We go from 1.5 million cases to 500,000 deaths per year, right? So we can actually treat breast cancer, but because it's so common, it still takes the lead in number of deaths. Other thing you should notice is cervical cancer, right here, cancer of the cervix. which I mentioned was often caused from HPV, that virus. So this is another reason women in particular should consider getting that Gardasil, that HPV vaccine, after, of course, you talk to your doctor. So once again, this should come as no surprise. The number one cancer around the world, also breast cancer. You can see how most of the Americas, Europe, all have the most common cancer being breast cancer. Interesting enough, some of these countries here have cervical cancer as the number one cancer as well. Okay, so what are some treatments? I want to spend a little time talking about different types of treatments to sort of help you understand what might be on the horizon, right? I should have started by saying so many people are affected by cancer. So in your lifetime, right, most people, it's estimated, will have a 1 in 4, 1 in 5 chance of developing some type of cancer. So treatment for cancer becomes important. That's why I think it's important for you to understand all this. So I'm going to go a little in depth into this. Try to latch onto as much as you can. So we'll start with just some general ideas for treatments. So once someone has a cancer diagnosis, what we want to do is we want to target rapidly dividing cells. Because after all, isn't that what cancer is? Rapidly dividing cells. Well, the number one ways we do that is radiation and chemotherapy. So I broke these down into our sort of main go-to treatments. We have local treatments and systemic, right? If you think systemic, that's going to affect the whole body system, right? Local, that's more localized to a particular area. So if you have a tumor, we might actually be able to go and surgically remove it. We can also send high doses of radiation to certain areas in a localized manner, okay? Then these more systemic treatments involved chemotherapy. We're going to go into a bunch of that in a minute, but if you think about it, chemical therapy, these are a bunch of drugs. Hormone therapy, we know that the hormonal system can be a leading contributor to some people's predilection or susceptibility to cancers. And then I'm going to spend some time talking about some targeted therapies that I think are novel and maybe going to make a big dent in our future treatment of cancer. Okay, so this is a difficult slide to explain. I included it to show you a few ways that the chemicals actually do their job, okay? So I told you for chemotherapy, we're going to use drugs that target rapidly dividing cells. Before you can understand this chart, you need to think back to your biology class and remember the central dogma, right? That our DNA codes for RNA, which codes for protein, which then ultimately all this together would lead to a cell. So if any of these are damaged, the DNA is damaged, has a mutation, the RNA is damaged, the protein, we get these cancerous cells. So when we have one that's rapidly dividing, it's just going through all this process all the time. So if we can stop any point in the process of making a cancerous cell, that would be good. So I want to talk about some of the big ones first. A lot of chemotherapies are taxanes that inhibit microtubules. So once again, think back to your biology class when we talk about mitosis and cell division. You may remember things called the spindle fibers. These are things that help move the chromosomes to the right direction. These make up part of the cytoskeleton. Well, these taxanes are inhibiting that cytoskeleton rearrangement, specifically the microtubules. So if we can stop the cell from being able to do mitosis, well, then it can't spread. Another interesting one that is commonly used are these here, okay? Camptothecins, etoposide, doxorubicin. These block an enzyme called topoisomerase. Once again, I'm going to ask you to think back to biology. Topoisomerase is the enzyme that unwinds DNA. So if you think about your DNA, right, it's really packed together, wound really tight and coiled. But for the DNA to go to RNA, right? We have to unwind the DNA, get to the genes so that we can copy them so that the cells can divide. So if we can block this DNA from even being opened or unwound, that's a powerful chemotherapeutic drug. There are also others that stop processes along the way, right? If we can stop the purines or part of the nucleotides from even being synthesized, that'd be great. Those drugs here, 6-mercapturine and Alameda methotrexate, you've probably heard about methotrexate in some commercials. They talk about that a lot. Another really interesting one are these kinase inhibitors right here at the end, because they block cell signaling to continue to grow. All right, so these are just a few of the ways that some of these chemotherapeutic drugs work. But if you think about this, if you're putting a drug in your body which is stopping cells from being made, not only are you going to stop the bad cells, the cancerous cells, but you're also going to stop the good cells that are rapidly dividing, right? Like our hair cells, some of our intestinal lining cells, some of the cells in our skin. So that's why treatments, the chemotherapy, are actually very taxing on people, because they're killing our good cells too. All right, so now I want to talk about a few novel biological therapies to sort of start to wrap this up. I call these novel because I think maybe this is where the future's going, because they're more individualized. Instead of like putting these chemotherapy drugs, which basically is like a bomb exploding in your body and it's going to kill everything, maybe we need some individualized, targeted, more like an assassin, to go in and just specifically focus on certain cancers. So I think this is the future of where we're going. So some novel therapy things we're looking at. One, monoclonal antibodies. And you've probably heard about this when we talked about COVID a lot. Monoclonal antibodies are just antibodies that we have cloned one of and made a bunch of copies that we know attack a cancer cell. So if you remember from your biology class how antibodies work, they basically log on to these little signal receptors on cells, whether it's a cancerous cell or some sort of bacteria or virus, and they do all sorts of things to kill it, right? They can signal for, or they can agglutinate and signal for cell apoptosis. But basically, antibodies help to identify and signal for destruction of a cancer cell. So if we know the particular cancer, we can develop an antibody to put into the cancer that can, or to put into the body that can detect those cancer cells and signal the body's own immune system to take care of it. Another thing we can do, or we're starting to be able to do, are some cancer vaccines, okay? So you may have heard, some conspiracy theories like, why isn't there a cancer vaccine yet, right? We made one for COVID in a year. Why can't we do a cancer vaccine? There's some conspiracy among scientists. All right, well, that's just ridiculous, right? There's no conspiracy among scientists. The problem is cancer's not one disease. It's many, 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 many different types of diseases. And so we can't just have one vaccine that kills all cancers, because every cancer, pancreatic, breast, lung, all of these are different cancers. So we have to treat them differently. And everybody's body is different, right? So one thing that we're starting to see development, or at least people are talking about, is a cancer vaccine, where what you do is you take an antigen. Now think about what an antigen is. Think back to your biology class. An antigen is a cell surface protein. Our cells have them, viral cells have them, cancer cells have them. It's basically a name tag for what's going on. So if we take a bunch of antigens, right, just the name tag of cancer, and put the name tag of cancer into our body, then the body will start to go through its own immune process. It'll say, uh-oh, I'm detecting some cancer. We need to mount a defense. So what it does is it will present that antigen, this is called an APC, or an antigen-presenting cell, to either a CD4 or CD8 T cell. Then those get activated, and then they present, right, to the tumorous cell, bind on, and then signal for destruction. Okay? So cancer vaccines, I think, are a possibility. I think we're going to see more and more of those develop. One that I think is just fascinating, this is the last one I'm going to talk about, is what we call CAR T-cell therapy. CAR stands for chimeric antigen receptor. And if you think of a chimera, maybe you've heard of this in like mythology, a chimera is sort of a combination of genes, or maybe a combination of some animal you've seen. So a CAR T-cell therapy, what we do is you have some T cells in your body, right? You've heard of B and T cells. What we do is we use a viral vector. We use a virus to insert the genetic material to make a receptor that then will bind to a target antigen on a cancer cell. So once again, let me explain that. We're taking some of our T cells from our body. We take them out of our body. We then add a virus which injects some genetic material. What genetic material? Oh, the genetic material to make this particular receptor. Why do we need that receptor? Oh, so that our T cells can find the cancerous cells because the receptor binds to the antigen on that specific cancer cell, which then leads to cancer cell death. Okay? So a lot of really cool things we can do here. As far as the last thing I've heard about these is that we're showing some remarkable progress with CAR T. So where is the future? I think these novel treatments are one way to do it. But the last thing I'll mention is the specific nature I think cancer treatment needs to move. One, if you think about how we do it conventionally, you've got all these cancer patients. We have one type or a few types of drugs we give everybody. Some, that drug has a very high effect based on their biology and genetics. Some, it has low or no drug effect. So in the future, I see us, and I think this is already happening, move to precision cancer therapy, where we take the patients, we do a genetic analysis of their particular genes that would tell us which treatment is going to be the best, so we can give different treatments to different cancer patients based on their genes or their genetic profile. So hopefully in this video, you learned a little bit more about the mechanisms of cancer. You learned some stats about cancer, and also maybe you're thinking about getting into research of cancer. Maybe you know someone and can advise them a little better or explain some of these things to them about cancer. It's a nasty, nasty disease, and hopefully science will help us progress to fighting this horrible thing.